Hello everyone, this is Tehsin and our team is here to explain you about protein structure and function in cystic fibrosis based on Maggie's illness case. Now let me introduce about Maggie's illness case first. Maggie was suffering from chronic wheezing cough for weeks. Dr. Jackson noted salt crystals on her skin and suspected to be suffering from cystic fibrosis. Cystic fibrosis is a genetic disease that affects more than 30,000 children, young adults in the US. This is caused by mutations in CFTR protein, leading to disease symptoms. Cystic fibrosis affects more organs but mainly lungs, pancreas, gastrointestinal tract. This leads to digestive and respiratory problems. One of the most common mutations in CFTR gene to cause cystic fibrosis occurs in exon 10, 1 and 524 nucleotides downstream of the start site of gene. Common mutations occur over 1000 nucleotide downstream of start site. Now, the mutation found in Maggie's DNA sequence will be explained by my friend Vanilla.
Mutation found in Maggie's DNA sequence. By observing Maggie's case, it has been noted that there is a mutation in Maggie's DNA sequence. The mutation is a single nucleotide substitution, that is point mutation, which have a variety of effects on downstream protein product. The change that has occurred in Maggie's CFTR protein is the substitution of one amino acid. In normal protein, the amino acid sequence is SFGR, while in protein it is IRGR. Now, function of CFTR protein will be explained by Kapila.
I'm Kapila. I'm going to explain the function of CFTR protein. CFTR protein is likely an integral membrane protein since it's depicted in relation to the cell membrane bilayer. CFTR protein helps to maintain the balance of salt and water on many surfaces in the body such as surface of the lung. The CFTR protein is a particular type of protein called an ion channel. Mutation in CFTR gene disrupt the function of chloride channel, preventing the chloride ions and water into and out of the cells. So now the loss of phenylalanine amino acid topic will be explained by my friend Vaishnavi.
Now I am going to explain the loss of phenylalanine amino acid. One small or large change in the sequence can completely change the structure of any protein. Therefore, loss of phenylalanine leads to the defective channel processing and getting problems. Loss of phenylalanine affecting the tertiary structure of a protein. The loss of phenylalanine can potentially affect the tertiary structure of the protein. Phenylalanine is a amino acid with an large aromatic side chains. Its presence or absence can influence the folding and stability of the protein. 3D structure is especially it plays the crucial role in the forming hydrophobic interaction, hydrogen bonds or other structural elements within the protein. Comparison of the normal and mutant letters of amino acid. Next topic explained by my friend Maria.
Now I am going to explain you about the role of water in cell with cystic fibrosis. In cystic fibrosis, the defective CFTR protein leads to thickened mucus affecting various organs. While CF primarily impacts the respiratory and digestive systems, it doesn't directly affect water regulation within cells. Water movement in and out of cells is primarily governed by osmosis and cellular processes, not the CFTR protein. Therefore, the presence of CF alone wouldn't necessarily cause water to exit cells.
Hello, this is Priyanka and
I'm Vidya. Now we will open CFTR normal protein structure and type IIFG, which stands for single letter amino acid abbreviation for isoleucine, phenylalanine and glycine. Now as you can notice, the yellow highlighted portion of the protein represent corresponding amino acid sequence IIFG.
and this is the CFTR mutant protein. We will highlight the amino acid abbreviation IIG, isoleucine and glycine. Now you can notice the yellow highlighted portion of the protein represent the corresponding amino acid sequence isoleucine and glycine. Now comparing the two CFTR normal protein on the right side of the slide and CFTR mutant protein on the left side of the slide, we'll compare the highlighted amino acid IIFG and IIG between them.
Again coming back to normal protein, changing to ball and stick style. Now this shows the structure of all amino acid, and you can see R group from another amino acid that is closest to phenylalanine ring. Moving on sequence alignment, you can find the yellow highlighted letter. One set of highlights is the IIFG sequence, originally choose amino acids. This is the IIFG sequence.
Now moving to the mutant protein and changing to ball and stick, we will also see the IIG protein highlighted area. Now moving on the sequence alignment, you can find the yellow highlighted letter. Onset highlighted is IIG sequence, originally chose amino acid. You can double click on II and G to view the exact amino acids represent on the ball and stick CN3D protein. Now we'll compare the position of R group between the mutant CFTR protein on the right side of the slide and normal CFTR protein on the left side on the slide to check the R group positions between them.
And you can see the R group positions are different between the normal and mutant protein.
This video is done with the help of CN3D protein viewer.
The conclusion of the topic is, when chloride no longer transported, there is a reduction of water leaving the cell. Cell water causes the mucus layer that lines R, GIT, RT become thick mucus, blocks small airway in lungs, difficult in breathing. Mucus traps are pathogens, causing large number of lung infections. Thick mucus coats the GIT, which prevents the secretion of digestive enzymes and absorption of nutrients. As Maggie grows, excessive weight loss and vitamin deficiency occurs. Now the MCQs will be asked by my friend Kapila.
Now let's see the understanding of the topic. MCQs. First question. Which of the following is true? Option A. Cystic fibrosis can be cured by medicine and doctors. Option B. Cystic fibrosis is a genetic disorder and no cure for cystic fibrosis, but range of treatment can help to control the symptoms according to the lifestyle. Option C. A is correct and B is incorrect. Option D. A is incorrect and B is correct. So the correct option is option B. Cystic fibrosis is a genetic disorder and no cure for cystic fibrosis, but range of treatment can help to control the symptoms according to the lifestyle. Second question. In Maggie's DNA, which type of DNA sequence is found? Option A. IRGR. Option B. SFGR. Option C. IGRG. Option D. SRGF. The correct option is option A. IRGR. Question 3. In loss of phenylalanine, which type of protein structure is attached? Option A. Primary. Option B. Secondary. Option C. Tertiary. And option D. Quaternary. The correct option is option C. Tertiary. Question 4. In cystic fibrosis, the defective CFTR protein leads to option A. Thickened mucus, option B. Lighter mucus, option C. Thickened mucus affecting various organs, option D. Damage of system. The correct option is option C. Thickened mucus affecting various organs. Question 5. Which type of mutation is found in DNA? Option A. Processing, option B. Frame shift, option C. Silent, option D. Nonsense. The correct option is option A. Processing. Question 6. What is the function of CFTR protein? Option A, maintain the balance of salt and water. Option B, breaks the salt balance and water. Option C, A and B are true. Option D, A and B are false. The correct option is option A, maintain the balance of salt and water. Thank you.